Hello, everyone. Welcome to today's live broadcast, Quantitative At-Line Portable Raman for Process Control, Development, Validation, and Implementation with a Pharmaceutical Example. I'm Laura Bush, the Editorial Director of Spectroscopy, and I'll be your moderator for today's event. We are delighted to bring you this web seminar presented by Spectroscopy and sponsored by B&W Tech. B&W Tech is an advanced instrumentation company that delivers lab-quality Raman spectroscopy solutions through user-friendly mobile platforms. B&W Tech provides solutions for the pharmaceutical, biomedical, physical, chemical, safety and security, and research communities. Their commitment to innovative solutions has made B&W Tech a leader in Raman spectroscopy solutions worldwide. We have a few housekeeping announcements before we get started. The webcast is intended to be interactive, so we encourage you to ask questions. You can submit questions at any time by typing them in the Q&A box, and you can find that by clicking on the red Q&A widget at the bottom of your window. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of your slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically. If you have any technical problems viewing or hearing the presentation, please click on the question mark Help widget at the bottom of your window. Please also note that there are valuable resources provided by our sponsor on the left-hand side of your screen in the green Resource widget. I would now like to introduce today's speakers. We are very pleased to be joined today by Michael Dotlich and Catherine Bakayev. Michael Dotlich is a research scientist in analytical sciences at Eli Lilly and Company. He works in the Lilly research laboratories validating test methods for clinical trial materials release. His active research is focused on the development of spectroscopic methods using different analytical techniques and chemometrics for identification and quantification of raw materials and drug products. He earned his MSc in applications of Raman spectroscopy from Marquette University in Milwaukee, Wisconsin. Catherine Bakayev is the Director of Analytical Services and Support for B&W Tech in Delaware. She has many years of industrial experience in the electronics, chemical, and pharmaceutical industries, with companies including GlaxoSmithKline, Camo Software, and Foss NIR Systems. Catherine earned her PhD in Polymer Science and Engineering from the University of Massachusetts at Amherst. She's the author of numerous articles and has edited a book on process analytical technology. She's been a member of the Society of Applied Spectroscopy since 1993 and served on the Executive Committee from 2010 to 2014. She serves on the editorial board of the journal Applied Spectroscopy and for NIR News. She's the past president of the Council for Near-Infrared Spectroscopy and a member of ASTM Committees E13 and E55. Thank you both for joining us today. Michael, you have the floor.
Thank you, Laura. I appreciate the introduction. As mentioned, this is an example of what we've used as far as chemometric modeling and Raman spectroscopy for an at-line testing of both Triton X and polysorbate 80. I also like to thank Richard Katner for the work he's done on this project. He's probably been the workhorse for the group in this area. Basically, Triton X and polysorbate 80 in the bioreactor type formulations are used for a couple different things. Triton X is used for clearing out a virus that could be in a bioreactor, and then polysorbate 80 is used for the drug formulation for stabilizing during freeze drying and storage and lyophilization. The solutions right now are prepared gravimetrically and for target concentrations. I'll talk about the target concentrations here in a little bit, but this is all done in a clean room. No further testing takes place once these are prepped. The engineer came to us and asked if it was possible for us to do testing in a relatively quick and fast and painless way for a user, in most cases an operator, for the testing. The instrument was going to be used by an operator. We looked at a couple of options. We looked at UV-Vis using solo VP. It's variable path length basically one would just enter the extinction coefficient and vary the path length to do linearity. Unfortunately, even with a variable path length, we had to get to a place that the operator was going to have to do some level of sample prep and reduce the concentration. Basically, we ended up with taking a look at Raman as an option for testing the material as it was prepared, which were, with the plans of doing no further sample preparation. So without further conversation around this, we'll just talk a little bit about the instrumentation. We did use, in this case, a BW Tech iRaman Plus instrument. Again, it's 785, 300 milliwatts. The software package was not one that we used that came with the instrument. We used a third party to develop a software package. As I mentioned, that we wanted this to be user-friendly. Part of the user-friendly preparation was, again, to eliminate any type of sample prep. Basically, the operator would take a sample of the material prepped gravimetrically and measure it, and the software that we wanted to use would be straightforward. In this case, I have a screenshot. You have a drop-down menu for the testing that would occur. Obviously, we do a QC test on a daily basis. The operator or analyst would pick Triton or Tween 80 as far as which material they'd be measuring. And then they click measure, and it'd measure and produce a result. And that result shown here, again, this is a QC check that we did on cyclohexane. It would show pass or fail, and then a second person verifier would verify the results. The QC test takes about 1 second. The runtime for Triton and Tween is about 20 seconds each. And the results, is I will demonstrate a little bit further, was predicted by partial least squares and chemometrics. Some of the challenges we were faced with is we wanted, again, as I try to keep this as simple as possible for the user. So we first approached this by looking at disposable polyethylene cuvettes, and that worked for a while. Unfortunately, we got enough of a difference in the backgrounds of the polyethylenes that we started running into modeling problems with background differences. I'll talk a little bit about the pre-treatment of the pre-processing of the spectra, but for all practical purposes, I didn't use second derivative, which probably eliminated this. But we stuck with a pretty simple pre-processing step. And in that case, we had to figure out a way to minimize the background differences between the empty or the different cuvettes that we were using that were polyethylene. We decided to take a look at quartz. The challenges with quartz, obviously, is the user would have to clean them on a regular basis, and they're not disposable, or they could be, but it'd be expensive, similar to what we would have with polyethylene. The polyethylene was such that there's enough of lot-to-lot differences between the cuvettes that we talked to the engineer and discussed the options that we had, and they felt it was more appropriate to go with a quartz cuvette and clean them on a regular basis. So we started out with a model on polyethylene, discovered the differences in the different lots and the background differences, and we decided to go with quartz after a number of attempts to correct the problem with differences between the polyethylene backgrounds. The calibration was pretty straightforward. It was over a number of days, and again, these days are listed sequentially, but in most cases it was probably over several weeks. Our target concentration for Triton was 25%. Our target concentration for Tween was 10%. We kind of took an approach that was similar to what you would expect for ICH guidelines of 80 to 120%. We got a little bit of curvature for Triton, so we narrowed it down from 23 to 27, around the 25, and then did pre-processing, which I mentioned before. I like to keep it simple with pre-processing. So we basically looked at the full spectral range, and then baseline corrected, and then normalized, and then obviously did mean centering. Again, it shows 11 days here. However, the 11 that are listed here could have been over several weeks. And so basically we captured as much as possible variability associated with sample prep days, and even included several lots of Triton to capture the variability into the model. The same thing occurred for, we took the same approach for Tween. Again, in this case, several days. And even in the case where we repeated some preps on both at the ends just to show there's reproducibility. So from this data, we generated calibration curves that we were planning to move forward with in a validation. One of the things I forgot to mention, but one of the practical sides of a portable instrument is you can do the validation in a laboratory setting and then pick up the instrument, and in our case a laptop, and carry it over to the manufacturing suite and do the method transfer. And so the examples I'm showing here are where we did the calibration in the laboratory, and then we did the method transfer in the production suite. So I'm showing here, as I mentioned, we did calibration. We got very good linearity. The C and CVs agree nicely. Very little prediction bias, or calibration bias, I should say. And the Q and T, hoteling T, are nice and fit nicely. So this is the model we moved forward, created a model for Triton and a model for Tween, and moved forward and decided to validate these two different models. We took an ICH approach to the validation, making sure that we captured linearity, accuracy, range, specificity, and precision. If you take a look at assay, it basically covers all those areas, as I just mentioned, even to the fact that we will capture specificity. I'll go through each one of these and cover them individually. Obviously, accuracy and linearity and range are captured in one when we did the recovery or the spike study. In effect, we are looking at more of a limit test. However, we took more of a holistic approach and did assay on this. Took it as more of an assay approach and did the validation and according to ICH guidelines around assay. So one of the things we started off with was linearity. We wanted to capture day-to-day variability and analyst variability and lot variability. So as you can see there, we looked at 5 different days, and we alternated between analysts on both Triton X 100 and polysorbate 80. From the validation data and some historical data, we were able to capture acceptance criteria for, in this case, we're looking at the root mean square error prediction of 0.42% for Triton, and then 0.36% for Tween 80. So basically, after we run our designed experiment, we should be able to meet these acceptance criteria. Shown below is the results of the study. I will say that there was partially my fault. There was a lack of communication, and the analyst basically did 4 preps on each of day 1, and we discovered that it wasn't necessary. They did one in the morning and afternoon, so we got a couple extra points, which isn't bad. A little bit extra variability, but instead of having a total of 5 runs in replicate, we got, I think, about 12. The bottom line was we got a fairly significant, the 3 root mean square error calibration and cross-validation and prediction agree nicely, around 0.1, as well as the same as this case for polysorbate 80. So that both were validated in the sense of we were able to show accuracy, linearity, and range. 3 of the ICH guideline points with the design study we set up for looking at several days and an analyst associated with doing the validation. Continuing down the path, looking at ICH guidelines, we looked at intermediate precision. Again, this is a design study. We looked at morning and afternoon for Triton X as well as Tween 80, and we alternated between analysts. So A1 would be analyst 1, A2 would be analyst 2, and they ran these on multiple days. And we're taking a look at nothing significant about morning and afternoon. It just made it made it convenient for us to divide up the work between morning and afternoon and define it as such. I think I made a mistake in the previous slide. This is where we ran the extra. They set up 2 in the morning and 2 in the afternoon, and they only needed to do 1 replicate, but they did 2 preps, so we got some extra data points in this run. So instead of the total of 8, we got 10. Nonetheless, we got extra variability associated with the run and capture to understand the method holistically as far as intermediate precision goes. So on the left, we show Triton. The average for all the runs collectively are 25, and then Tween is about 10%. Repeatability had an acceptance criteria of less than 0.16 for Triton, and tween 0.12, and intermediate precision, which is a combination of within run and between run, had 0.23 and 0.22, about the same. But yet we clearly met those acceptance criteria in both cases. For Triton and tween, for Triton we got repeatability of 0.03 and then intermediate precision 0.04. Tween was 0.04 and 0.13. So meeting the acceptance criteria for intermediate precision, we felt comfortable, and we continued on with the validation. And we began to look at, in this case, we wanted to decide what method acceptance criteria we'd look at to determine what we would consider to be outliers, as you can imagine with chemometric modeling. And the best example I'd like to share with this is you have an HPLC test. We typically have system suitability related to repeatability. So, for example, a method may say 5 standards prepped and ran up front should have a RSD of a certain number in order to move forward with the test. And then in some cases for rel subs, you'll see acceptance criteria around resolution. In the same token, we wanted to do the same thing. We wanted to have a method system suitability acceptance criteria. And part of that challenge was, how do you do that in a chemometric model? Well, we had to first figure out what kind of things would go wrong, and from that decide what items we would focus on and how we would capture those. For example, how would we know the wrong type of sample was, or that the analyst picked the right method? For example, if they prepared Triton in the clean room and they picked a method for tween, would we be able to capture that? What if there was an empty cuvette they grabbed and put it in there and for some odd reason forgot to fill it? How would we catch that? Or what if they filled it with water and forgot that they had water in it and put it in there and ran it? What would that look like? And then also, what does laser power do? And then X shift, if the x-axis were to shift a little bit, how does that impact the results? So we kind of looked at all those. The first 3, I call them no-brainers. Those are going to have high Q and T, and I'll show those a little bit. So Q and T would be very large. Laser power, we basically tackled with our cyclohexane QC test that we run at the beginning of every run, and I'll show some details around that. And then the x-axis becomes a little bit more involved. From the data that we've generated to date, we knew that we could handle an acceptance criteria with a reduced Q of about 7 and a reduced T squared of 10. And then that's for Triton, and for tween it'd be 20 and 2 respectively. So those were the target values. So anything greater than those we'd consider to be outliers, and we knew that because we looked at the data we generated so far and said, hey, all this data is good. And then we generated some data with, for example, the polyethylene. And the polyethylene stood out as an outlier, so we drew the line between what we knew to be good data and bad data and picked those limits. I mentioned laser power would be captured by cyclohexane. From these acceptance criteria and known variability in the method validation that I showed previously for linearity and intermediate precision, we said, what is an acceptable X shift that will allow us to continue to perform within a reasonable level for a method? In this case, the method was the model. So what we've seen historically. So first I want to say, okay, what have we seen historically for system suitability? So cyclohexane, during the course of developing both the calibration model and doing the validation, we generated quite a number of cyclohexane runs. And I noticed that for all practical purposes, the highest peak was around, and this is in pixels, around 456. However, there was a small shoulder at 457, and in some cases, some of the runs did not show that shoulder. So it kind of showed me that there may be a shift of a peak, and if you just extrapolate or infer where it might be, it could be anywhere between 456 and 457. So I said, we're probably looking at about a half, 0.4 to 0.5 wave number shift, 0.4 to 0.5 pixel shift, which is equivalent about one wave number. And again, how does this impact? What is this shift? How does it impact the model? All right. So first, our goal was, I showed during the method calibration, not calibration, but the validation, but looking at linearity, that for Triton, we had about a root mean square error of about 0.04. our goal was, we need to stay as low as we can on the root mean square error, or in the case of intermediate precision, which was bigger, we'd use that number. But in this case, I went with a smaller number. So the root mean square error that we got out of linearity was 0.04%. So if I said, okay, our relative error, known error, assuming it was all related to pixel shifting, and this is pretty conservative, is about 0.04%, and I'm not sure if these are hard to see from your end or a little bit for me, but the number turns out to be, so if you look at about a 0.4% based on the cyclohexane, the previous slide, we get about a 0.04 relative error. And then over on this side, we get a little bit less than 0.04 relative error. That translates into an absolute error of around as I recall, about 0.1. And that's pretty reasonable. So that means that my error for this model would be plus or minus 0.1 if I have a shift of 0.4 wave numbers, pixels. How does that impact T? So remember our acceptance criteria for Q and T is 7 and 10. So for T, I want to be less than 10. So if I have a pixel shift of 0.4, I was getting just under 10. So that should work. So I'm good with that. So a pixel shift would result in a 0.4. A pixel shift of 0.4 would cause me to have a less than 10 T squared. The same with Q. Reduced Q is the limit of 7, and so if I have a pixel shift of 0.4, I'm going to be well below that. So in all cases, pixel shift of 0.4 will work for the Triton X method, giving me a very small amount of error in the result, and it still passes my acceptance criteria. So what does it look like for tween? So for tween, the root mean square was 0.1, which was pretty consistent. The root mean square was 0.1 for the accuracy, as well as it was 0.1 for the intermediate precision. So I really didn't have to choose between this one. But in this case, if we go with a 0.4 pixel shift, we're right at 0.1, which is reasonable. The absolute error was going to be, I think it's less than, the absolute error is going to be at a 0.4 is going to be less than 0. On the high end will be around, it's going to be around 0.1. So 0.1 plus or minus 0.1 around my result is going to be reasonable. So for example, if it's 10, I could be 9.9 or 10.1. And just to remind you, it was on one of the slides that spec is 9.5 to 10.5 for tween. For Triton, it's 24.5 and 25.5. That's the range we need to be on as far as reporting values. Again, go and look at impact on T-square. A 0.4 shift plus or minus is going to be less than the acceptance criteria for T, which is right around 2. And then for Q, my acceptance criteria is going to be 20, which is way up in this region, and we're well below that. So again, 0.4 works for both models and for purposes of what we'd expect for cyclohexane. So what does that mean for cyclohexane? So we use HQI for our determination. So the analyst comes in, runs cyclohexane as a system suitability check, make sure the system's running properly. Well, what would we expect for an HQI? Well, if we look at the data, and we're at pixel shifts of 0.4 plus or minus 0.4, the range could be anywhere between 96 to 99. I conservatively said let's just round down. I said we'll go with 95% HQI. And that seemed to do the trick. We've not failed to date on that HQI. However, we have had some challenges with, I think I discussed next. I will talk about it later, but we'll look at laser power and the impact on that. But we had some user error around laser power, and we found that we were able to capture some of the failures. So specificity. So we know what our acceptance criteria is around Q and T, and pixel shift, and what if they make sense. So I switched the numbers here because it's relative to the graph. The ones were reduced Q. In this case, we're looking at absolute. They're the same number. It's just one's divided by this 95% confidence interval. This is not divided by the 95% confidence interval. But we looked at specificity. I mentioned before, what if we ran an empty cuvette? What if they prepped the material at the wrong concentration? What if they grab the Triton and they prep it at 10% thinking they have tween? What would the result look like? Triton at 100%. What is Triton at 25%? That's my control. It obviously passes. Tween at 10%. So the fail there is that they ran tween on a Triton method, and we caught it. Tween at 100 and tween at 25%. So tween at 25% would mean that they ran tween thinking they had Triton prepped at 25%. Obviously, it fails a high Q. So we're able to capture any type of improperly prepared material when running the Triton method, and the converse is true as well. Empty cuvette, what if they prepped Triton instead of tween and they ran it? Well, it'd fail at the high Q. What if they prepared, well, if you ran Triton at 100%, that's more of a control to show it fail. And then Triton at 25%, what if they ran the wrong method? So we prepped Triton and they ran they prepped Triton at 25% and ran it on tween. We'd fail. Of course, the controls passes and so forth and so on. And obviously water, if they ran water, it'd fail. So the specificity is very discriminating between the sample. So if the model is capable of determining whether or not you ran the right material. So I also talked a little bit, what if there's user error? So we noticed that on the particular instrument we're using, that the analyst, if they didn't insert the laser probe completely into the cuvette holder, that there might be a gap. So what does that gap do? Well, it turns out if there's a 1 or 2 millimeter gap, for Triton at least, the limit, again, this is the Q limit, if they go to 3 millimeters, they'll go above the limit and fail. This case wasn't the same for polysorbate 80. They could tolerate about a 3 millimeter limit. Even 4, which isn't showed here, does fail. It's off scale. I just decided not to show it because I'd have to zoom out quite far. We also looked at cable position. So fiber cable is pretty susceptible to positioning and its impact at throughput. So we just wanted to make sure that if the analysts were moving the cable around to reposition it, it wouldn't have any impact. And so we showed that very little impact on both Triton and tween. We also looked at fill level. What if the analyst forgot to fill it completely? Well, to catch it. In this case, this fill level is at laser, so the fill level is right at where the laser hits. It failed. So we just know it wasn't full. And then, it doesn't matter if it has a lid on it, no. And then the control here is repeatability. We just looked at the multiple runs of the same thing at method conditions, and obviously it passed at this point down in here. So for all practical purposes, ruggedness can be defined into the method, and we can capture when it's being run accurately and consistently. So here's where we talk a little bit about laser power. I just start. The cyclohexane, our QC check, it fails at 75% threshold. In all these cases, we went from 75, 15, 25. In all cases, they passed the model, and so I just looked at repeatability. So 125 down to, 100 down to 25, we get results of consistently across the board of 25.2, 25.1, 3, and then 10.4. There was really no impact to laser power. Basically, the bottom line is laser power has no impact on the result. However, we would catch it with our cyclohexane check at a 75% threshold. Robustness. We're going to look at probes. So what if the probe we had broke? Can we introduce a new one? Well, we could, and I'll talk a little bit about that. It's a lot easier if you include a calibration of that second probe into the model. In the case we show here is you just take that original calibration and you run that second probe, and we get a little bit of a bias with the result is by including the second probe, by running a second probe. The same is true for PS80. However, if we include the second probe, the bias goes away. Get a little bit of a skew in the case of PS80. But you can see here that the results are somewhat biased, high. This is the absolute value or the measured value, prepared value. And then the predicted values, once we included the second probe, were consistent with the measured. Same is true for tween. However, you do see a little bit of a skew here. We hit it in the sense that it's not predicting completely accurately. Not terribly different between the two, but enough that you'd say, let's include the probe in the calibration. So if you had a new probe, you'd have to include it. But one of the things we're looking at, and I think I talk about it here in this next slide, what if we included a probe up front? One of our customers wanted to have redundancy, so we're going to end up providing with them with one instrument and two probes. So part of that redundancy is what do we do for them to connect two probes to it, and well, not simultaneously, but being able to in the case that one probe goes bad, how do they change it out? And we can provide some assurance that the results are going to be consistent, because in this case it's going to be in a routine area, and they're not going to be in a place to update the model. So we'd like to have this done in advance. So what we did is a little bit of work with Catherine, and this is looking at a way to normalize the data. So we I'll talk a little bit about that in a second, but this is not corrected data. We'll talk a little bit about corrected data in a second. But in this case, we're looking at the two probes separately. So in other words, this is instrument 1, and this is for the sake of comparison. But in instrument 2, we had two different probes. In this case, each probe was run independent of one another and predicted independent of one another. In this case, we combined the two probes and ran them. I'm sorry, this is for tween. We combined the two probes, and we got accurate results. Tween behaved a lot better than Triton in this case when we used the two-probe model. As you can see, there's a little bit of a bias when we combine the two probes into one model and predict the results. In the case of tween, we weren't so concerned, but we said, how do we address the slight bias for Triton and when we combine two models into one probe? So this is the previous slide showed without instrument 1. I just zoomed in to kind of magnify the bias. 25 is our acceptance, or the target value. You can see one was slightly low. The mean, my apologies, the mean is about 25, and the other one's just a tad bit high, so it's 25.1. And I would have to say some may not even consider this a significant difference. However, my curiosity got the best of me, so I wanted to figure out a way if we can make these more alike. So this is where we spoke with Catherine. We normalized the data using we basically did a normalization of the background between the two probes. And we came back with the bias is gone. We're getting numbers very similar, 25.1 and 25, but for practical purposes, it's very similar. The downside is we increased the variability. So our standard deviation went from 0.05, 0.07 to 0.1, practically speaking, for both of them. So the probes look much similar, but we introduced a little bit extra variability with the correction, doing intensity correction between the two probes on the same instrument. So the decision to go forward, and I think I talked about this here in this next slide, was to go ahead and let's continue this intensity correction to see what it gets us long term. So we took, again, here's instrument 1 just for reference, because this is the instrument that's been validated in manufacturing right now. This is the instrument that we're looking at for transfer to another customer. Instrument 2, again, with two different probes. So this would be probe 1, and this would be probe 2. The labeling, we use a little bit different label. We called it B1 for backup 1 and probe 2 for instrument 2. But again, this is two different probes on instrument 2. We took a look at 12 consecutive, we took a look at several months, I think this is about, a month and a half of, about a month's worth of data over several days. We did 2 preps per setup per day, and approximately 16 to 17 samples, and ran them on instrument 1, 2, probe 1 and 2 on instrument 2, just to compare. We introduced, on instrument 1, again, this is just one probe. On instrument 2, we have 2 different probes, and we did intensity correction. And we got decent results, even similar to what we'd expect from, and again, I showed the limits here of 24.5 to 25.5 just to show how well we produced the results. But both probes behave quite nicely between the two instruments. If you look at setup and within, so most of the variability on probe 1 on instrument 2 is more within run. Is basically setup and within run for probe 2 is consistent. But the total intermediate precision between the two instruments is similar, and I think it was around 0.1 for both of them. So I also just went, for the sake of comparison, this is historical data on instrument 1. The intermediate precision over the course of a year was around 0.4, which is quite good. Again, I'm a little bit tighter than the limits, but none of the data failed. The point of this slide is to show performance over a year's period of time. And the fact that we did a model adjustment, and by changing the pre-processing, we tightened up the variability of the method. So how about model maintenance? as I mentioned, instrument 1 was transferred over to manufacturing, and part of that transfer, we noticed that there was a bias that we began to show in the original data. So in the original model, we were getting a bias, not much, by about 0.1, and I thought, what can we do to make this better? So what we decided was to add in I added in the validation data from this method, and once we did the transfer, to see if we couldn't increase or reduce the bias, and of course we did. So part of model maintenance is, in this case, to add in the validation data if you can. And the nice thing about adding in validation data is it was done under protocol, and quality is signed off on it. Therefore, when I add it in, it's gone through the rigor of the quality process, and so adding it in isn't something I'm doing. It's something I'm doing under procedure and protocol, which makes it, I suppose, more acceptable from a quality perspective, and it makes it more easy. It makes it easy to explain if it's being reviewed by a regulatory agency by adding in known data associated with the process. So, if we were to add in future data, I would suggest we'll probably do that under protocol as well. But that was a convenient time to do the correction to the bias. So just a little bit more on intensity correction here. In this case, we had two different probes again, and we were looking at instrument maintenance. I mentioned that we had some user error in one of the instrumentation. What happened was the metal filing. We had switched the probes around a number of times, and we'd not cleaned off the connectors, and some metal filings fell into the fiber, and they burned and etched into the fiber, so we had to send it back for repair. So this uncorrected data shows after repair, and it shows probe 1 and 2. Again, I call it's probe 2 as we define them here, and probe B1, but again, that's backup 1, so it's probe 1 and 2. Quite high in the hoteling T and Q, and I think they passed, but barely. So it made me think, well, what can we do to get this back in line? So we used intensity correction, and it brought everything Q and T back in nicely. So again, it shows that with intensity correction, you're able to correct for some of the differences associated with the modeling before and after maintenance if you were to get into a similar situation that we might have been in. So in conclusion, calibration, if done correctly, you can capture quite a bit of variability associated with material lot, the quality, analyst, and instrument. Validation parameters ensure a robust method, and especially if you follow ICH. It gives you a nice little guideline to follow through. Appropriately established Q and hoteling T acceptance criteria are great for using as a system suitability. And then I mentioned instrument portability gives the ability to do the method validation in a laboratory and then transfer to a manufacturing site. Ashley and Brady did a lot of the lab work. This is one of our customers that we're working with overseas. This is the engineer that came to us to ask, hey, can we do this testing? And then both Bob and Jeremy have been perfect in providing a lot of solutions for us for the chemometric model as well as the front end for doing the testing. And I think that is it, and I'll pass it over to you, Catherine.
Great. Thanks very much, Michael. And everyone, just to let you know, we'll be taking questions at the end, but I encourage you to type in your questions now that you may have for Michael and any that may come up as I present just a little taste of some other things that one can do for process monitoring with our portable Raman instrumentation. As Michael mentioned, he's used the iRaman Plus, which is a nice portable Raman instrument that can be used for both qualitative and quantitative analysis, and we have softwares that are specialized for both of these, as well as the ability, as Michael and the team at Eli Lilly have done in developing an interface that suits their final needs. We use this in many different types of applications. For process, I'll show, again, brief examples of reaction monitoring, polymorph transitions, and solvent recovery. We've also used this in blending, in formulation work, and so on. The portable size of this instrumentation makes it ideal for work such as Michael's team has done in doing the development work within their lab and then taking that same instrument and moving it into that final manufacturing suite where they want to be making the measurements. So the portability brings great advantages in the ability to do the work where it's needed. And we do have customers as well who do the same types of reactions and processes in different locations, so they want to be able to just take that same instrument to different places. We do have both lab and industrial grade probes as well as flow cells for various sampling environments. In the case of what Michael's presented, he's used our lab grade probe and interfaced that to a cuvette holder. So rather than going directly in situ for the measurements, making his measurements with the probe interfaced to a specialized cuvette holder. So their samples are well controlled in the quartz cuvette, as he's mentioned. Our next generation of instrument, the Raman Pro, brings even greater benefits in having deeper cooling CCD detectors, which give superior performance for long integration times and low Raman scattering materials. In this instrument, we have now an integrated tablet PC with our BW Spec Mobile. So you have even increased portability with this because you don't need to also port a computer with you. With this as well, we have various softwares that can be used with it, with a computer if that's the preference, and a sampling with the fiber optic probe covering the spectral range from 65 to 3200 wave numbers when using our 785 nanometer laser excitation for this system. Now to just show brief highlights of a few examples that we've been working on. This is some very recent data that we've been working on for some in situ reaction monitoring for the synthesis of an active pharmaceutical ingredient. And we are still waiting for some reference values to go ahead and do the quantitative analysis of this. But this reaction is carried out in acetonitrile. So we have some of the strong peaks of acetonitrile present here. But more importantly, we can very clearly see the product peaks here that are growing in over the course of this reaction that was run for just over an hour. And likewise, though it's maybe a little difficult to see, and actually my arrow has moved over, I do have a starting material peak here that is shifted over basically as it's diminished and the product peaks have come in. So this is just to show you the power of using the portable instrumentation for reaction monitoring. And in this case, what we did use is our immersion optics. So this was directly inserted into the reaction, which was run at reflux in the acetonitrile. So it is a direct in situ measurement in that way. Another example is here in using the Raman to study a polymorphic transition. Now, polymorphs are, of course, very important in that drugs can show different solubilities and different efficacies depending on which polymorph is present. Citric acid is a nice model system because we know that when it's formed by crystallizing cold water, we have the monohydrate form, and then we can form the anhydrous form by crystallizing at higher temperatures and readily convert from the monohydrate to the anhydrous form by heating this system. So here I have the Raman spectra of the two forms overlaid with each other. One of the great things about Raman is it's very sensitive to local bonding and molecular arrangement, so the Raman spectra are quite different from one polymorph to another. Here's showing expanded views of the different forms, the monohydrate and the anhydrous form in both the OH region and CH region, where we can see that we have, as we go from monohydrate, we lose the peak here at 1108, and we have a peak that grows in here. So we can use this then to monitor the transformation as we heat from the one form to another. So in this case, again, we were doing this with, it is a solid form, so we're making the measurement with our probe focused onto the solid as we're making the heating it and doing the transition. And here we're using software, our BWSP software, that allows us to do that real-time monitoring. So here I have an overlay of the spectra, and here I have basically the transition from the one form to the other as we're following the peak from the monohydrate to the anhydrous formation. And we have the option here as well to do quantitative monitoring, but in this case, you know, capturing a sample and getting quantitative results wasn't feasible, so we used some some PCA modeling, and in this example, a simple monitoring of a single peak related to the form in question. In this other example, what we're looking at is basically a solvent system where we're looking to see how well we can quantify low levels of acetone in a mixture of other solvents. So again, we're using an immersion probe inserted into a glass reactor as we're doing gravimetric changes in the additions of the solvents. So here I have an overlay of the solvents that we're measuring, and the acetone is what we're interested in. So I've highlighted the very strong peak of acetone here at 786 and another one here at 1709. The other solvents, the TAF, ethanol, and methanol, also have peaks, and here we have just this very nice clear separation of this acetone peak. So when we make the addition of acetone into the mixture, it's hard to see in this raw data, but we can clearly see how the signal of that acetone increases as the concentration increases. And here we go forward and develop a chemometric model based on partial least square regression on the raw data to show that we can readily quantitate the level of acetone. So this is showing me my predicted versus measured values for acetone with calibration and cross-validation, validation samples in blue and red respectively, where we can see that we have a root mean square error of less than 0.01% of acetone here as we span this narrow range of acetone concentrations in the mixture. Then using this model then to predict on independent samples, we see again a very good linear fit for the prediction of samples that were not included in the calibration model. So this is showing you that this portable instrumentation can give you very good, strong results even at the low concentration levels, such as shown here. So my point today was to reiterate, based on a few other examples in addition to what Michael's done, that portable Raman is an excellent tool to increase operational capabilities and reduce costs while providing solutions from rapid material identification to process understanding and real-time analysis where you need it. So by the portability of your instruments, you can take your measurement to your samples and not vice versa. I'd like to thank you all for your attention, and we will be having time for questions right now. But for those of you who may have more specific questions that you'd like us to address, here are mine and Michael's contact information. And with that, I'd like to turn it over to Laura.
Thank you very much, Catherine, and Michael as well for that excellent presentation. It is now time for questions from the audience, so let's get started. But I just want to remind everybody that if you'd like to ask a question, you can type it in the Q&A box, and you can find that by clicking on the red Q&A widget at the bottom of your window. Okay, first question, Catherine, this one's for you. How do you insert a Raman system into an online or at-line process?
Well, I think Michael showed a very good example of how one can use it in an at-line process where they've developed their methodologies within the laboratory, and now they're moving into a clean room where they have procedures and protocols in place where they'll be taking the samples that are prepared using the cuvettes and making the measurement. Similar things can be done depending on what the process is, where one can develop the calibrations in the laboratory and transport the equipment to where the measurements need to be made. Some of the questions or decisions that users need to define is how do they want to interface that? So will it be by taking samples and measuring them in cuvettes or perhaps disposable glass vials? Or will they make an interface such that the probe is inserted into the process or into a recycle line, so not directly into the vessel itself of the process, but perhaps something that is taking sample and reflux or recirculating it around. So it will depend on the process, but clearly we can do this in many different ways.
Excellent. Michael, has this method been tested with buffer species, other excipients, or protein in the solution with the tween?
No, it has not. We've looked strictly in this case at tween 80 and Triton in the clean room preparation prior to dispensing into the bioreactor or the formulation. So basically this is a pretty straightforward test. We're looking at the prepared raw material at concentration with no other components in the solution other than water and tween or Triton. We have looked at, in another approach, we've looked at bioreactor materials. The piece with Triton and tween is that Triton is fairly low concentration, and we're likely not going to see it in the bioreactors themselves. In the same case for tween, some of the formulations, they're fairly low in concentration, and I don't think we're going to be able to see them. At least we haven't tried. We've looked at more of the more common, when we're looking at the complex matrix of a bioreactor, we're looking at more of the higher concentration items, such as a titer for the protein or glucose or titrate, or glucose or lactate.
Okay. For Michael or Catherine, here's a question. For establishing a Raman validation method for raw material used in manufacturing, is it required to first go through the calibration before starting to test samples?
You want that one, Catherine, or you want me to answer?
I'll address it, and of course welcome your input as well. If I understand this correctly, the question is whether or not methodologies for validating testing for raw materials are needed before going through and developing a calibration, much as Michael has done. There are requirements for incoming raw material testing, and if one is using, when one is using Raman for this, yes, the methods to do this do need to be validated. So that would be typically a qualitative measurement, and it may be done with a handheld or a portable instrument, and that would be a separate step in the analysis from doing quantitative calibration, such as done here in prepping materials to go into the process. But again, I'd like to hear Michael's view on this as well.
Yeah, thanks, Catherine. I think that you were consistent with what I was going to answer. The regulatory requirements require some level of validation for release of material for identification by, in this case, Raman. If it's a simple validation, if it's a simple ID method, and it's for release, the validation can be show specificity of that material. In the case of some of the Raman instruments out there today, that specificity would be against the library that's established, and then demonstrate that you can run that material against that library and it picks that material. In the case of doing some of the requirements associated with the Pharmure and around 100% identification of the different drums that are maybe in the warehouse, it's important to do the same steps, but because that's not for release, it may have a little bit lower requirements. However, we've considered it to be one and the same, and we go take a holistic approach and say, hey, we're going to validate this method for identity, qualitative identity of the material against the library. In the case of the example I have here, the validation is required because technically I need to have some level of confidence that the calibration is going to work when I'm predicting a result. So that requirement is more of a technical requirement, not necessarily a regulatory requirement.
Michael, were you able to transfer a model from one instrument to a second one? And if so, what was the accuracy and variability of that?
At this point, we haven't been successful at doing that transfer, and that's in part because we only got one instrument up and running. I will say that we've done a number of tests across the instrument platforms of taking the model and transferring over to a different instrument, and without the intensity correction, we've been unsuccessful. I have just now gotten into the place that we were going to use intensity correction between instruments to see if that works. So at this point, no, but I'm hopeful that we could with intensity correction.
And can you explain the intensity correction process?
Yeah, it's quite interesting. I think within the past several years, the NIST came out with a green, and I'm not even sure it's green glass. I believe it has some level of nickel in it. You order it, and actually BW Tech has a manual on this and how you do this work, and they have a nice little setup. But you're basically generating a file that does, well, you insert the green glass into the cuvette holder, and they have a procedure in their manual to follow to in order to generate this intensity correction. You follow the manual and procedure. You generate a file that has values associated with the intensity correction for that probe and detector. So the probe and detector are unique to the intensity correction. And then that file you can turn on during the testing to do the correction. And then basically the hopes there is that between probes you would be able to mitigate any variability associated with significant intensity differences related to laser power or detector response.
Excellent. Catherine, what kind of support is available for setting up an online process?
At B&W Tech, we have the full support in both the method development and validation, as well as implementing systems into a system. Now, sometimes it will also, oftentimes I should say, it will require some engineering support, which is best served by the engineers at a site where things will be implemented. But in the case of something as Michael is doing, where it's an at-line implementation and it's a matter of putting the instrumentation near the point of manufacture, we would offer the full support from qualifying the instruments to working with the calibration modeling as all the way through to the validation of the methods as Michael and his team have done on their own, and transfer of calibration to other instruments.
Catherine, does the portable iRaman Plus have different excitation wavelengths other than 785 nanometers?
Yeah, the iRaman Plus is available also in 532 and 1064 wavelength excitation. So for samples that are highly fluorescing, 1064 is more appropriate, the longer wavelength. But yeah, the iRaman Plus does come in three different wavelength excitations.
Fantastic. All right, well, I think it's time to wrap up. Catherine and Michael, thank you so much for your excellent talks today and for answering those questions.
All right, thank you.
Thank you, Laura.
I'd also like to thank all of you in the audience for watching today and for participating by asking questions. And I'd like to thank our sponsor, B&W Tech, for making today's webcast possible. Please note that this webcast will be available for on-demand viewing through June of next year. You will receive an email message from Spectroscopy alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who might have missed today's live event. We hope to see you all next time. Goodbye.